what are the challenges or the new things which industry is trying to pick up, and where industry and academics can actually collaborate and go forward for. Before going for the presentation, I have a declaration saying that this is purely my own presentation, and whatever I have been discussing, I'll present my views, and it is only for knowledge purpose. It has nothing to the place Unichem where I work, and it doesn't require any legal obligations to it. To start with, I'll have a quick introduction about the lipid-based delivery system in general, then a few critical steps in development which we as a formulator and industry face. What were the major challenges in that critical step that we tried to solve? A few limitations of this delivery system, and a very good case study from our industry perspective wherein the product was developed and launched in the market, and references at the last. To start with a brief introduction, as you know, the oral delivery is one of the most preferred delivery because it's the natural route of administration. There is no unagitated need for that. With the increase in focus, there are a number of NCs which comes into molecule and becomes active. Nowadays, even BLEs, biologicals are also into the market very quickly. So both for small molecules, NCs and biological molecules, lipid delivery system is one of the key factors. And that is the reason all of the industry focus is now started to shift from normal conventional oral dosage form to more advanced lipid-based delivery systems. And when we move to a more advanced system, the more complications do occur and more difficult challenges we need to sort in the industry. To give a brief, so the formulation technique which we use currently to improve viability of a very molecule which are notorious for rock-like salt, there are many techniques which are already been used. The list which I have given is in the sequence in which industry actually try to improve a viability. The first and simplest thing is to add a pH modulator in your formulation if the drug is showing a pH dependent solubility, so that it improves its solubility and the viability in turn is increased. In case drug has a precipitation tendency or what we call an Oswald ripening, once it gets solubilized and comes out, we use precipitation inhibitors. Majorly, a polymer systems are used for this purpose. If both this technique doesn't work, then go for a little more deep into the drug molecule wherein you micronize, you reduce the size, increase the surface area of the drug and get the drug solubilized so that the viability may increase. Salt formation is an alternate, but salt formation is a fourth choice because it changes the entire chemical nature of the molecule. So if the first three types doesn't work, then you move to the salt formation. Solid solutions and surfactant addition are next stages of solubility enhancement and in terms of viability. Nanoization and complexation are more advanced. There are many steps of characterization which are required and the development time get increases. So these are more complicated systems when it is done and less preferred in terms of costing in terms of industries. Polymer dispersions, amorphization and eutectic mixture are also one of the key parameters, but you require a specialized instrument to make these type of systems. So you require a dedicated line of system in the industry and the commercial lines, which make them less viable for industry, or only few of the organization does that. Coming to that, lipid-based delivery system is what is growing up now because it doesn't require a heavy investment in terms of getting some instruments. Normal beaker, stirrer or homogenizer with a proper addition mixing can lead to a development of a lipid-based delivery system. Just to recap, the type of lipid-based systems which are, there are almost like 4 major systems classified. I've taken it from a literature, references are at the last. The type 1 system is wherein only the oil component is used and we don't use any of the surfactant. There are only 2 products in market currently as on date approved using this system. There are filled in oil, drug is dispersed in oil and filled in capsule administered to the patient. Type 2 system, this is a category wherein mixture of oil and water insoluble surfactant is used. Herein the surfactant concentration is also very less. This is secondary type wherein pure oil dispersion of drug is difficult. Drug is not neither hydrophobic nor lipophilic. So in that case, you require a surfactant which is insoluble in water to disperse the drug. That's when where you use the type 2 system. Then we move to the more readily used available system, and lots of literature will work around system 3 and 4. 3 is again divided into two parts, 3A and 3B, wherein 3A is a system which generally consists of oil components and have a range of 40 to 80% of oil. The surfactant here as compared to type 2 is a water soluble surfactant, and HLB is more than 12. That you can use a co-solvent or you cannot use a co-solvent depending on the formulation type. Wherein type 3B, you have oil component which is very less, which is less than 20%, and the surfactant and soluble components are more in quantity. The difference in type 3A and 3B is type 3A is the system which generally used for a low dose drug which is more soluble in oil. Type 3B system is used for a low soluble drug which is more soluble in combination of oil and surfactant. So during the initial screening of the solubility of the system, we generally define which type of system 3A or 3B to be manufactured or go forward for development. Type 4 systems are generally oil free. So here the molecule per se is not lipophilic, it is just hydrophobic in nature. So just to remove that hydrophobicity, what you do is you remove the surface, the overall difference in reduce the surface tension. When you reduce the surface tension, you require surfactants. So you add lots of surfactant to the component, and with that combination of water soluble or insoluble component surfactant along with solvent can give you a type 4 system. Type 4 system are usually less preferred because they crash out very easily. They once you add them in water or when you administer them to the patient, they immediately crash out into the GI system, and the overall effect which you see in vitro doesn't turn out in vivo. To go more deep down into what happens when you administer it to a patient in terms of LBF is the the slide is divided in three sections: the left, middle, and the right. The left section is about where we can actually modulate, wherein you orally administered a lipid-based delivery system, and in stomach with the help of lipase and gastric linguids, it get digested into glyceride, triglyceride, fatty acid, and free drug. The objective here is to minimize the quantity of free drug and maximize the quantity of tri and diglyceride which are in combination with drug. So free drug will lead to loss of drug, and only when you make this micellar systems, that's when the maximum absorption would happen. When this system moves to the small intestine, so there three components are we majorly look is the shear force, the gastric emptying time, and the activity of pancreatic lipase and colipase. That's where you get the emulsified monoglycerides and fatty acids, which in turn form multi-layer vesicles, unilayer vesicles, depending on the amount of lipid, types of lipid, and vesicle natures. And if you see the component, there again a free drug is formed. That free drug doesn't crosses the enterocyte and the lymphatic system. So that is the drug which is lost. So the overall objective here is to minimize the formation of this free drug. So the more effective, so this is where the type 4 system fails the most because they immediately crash out and let lot of drug free. 3A and 3B types of lipid systems are majorly helpful in minimizing this free drug component. So the other part which is formed like multilamellar or unilamellar vesicles, they convert themselves into mixed micelles. That is what it can cause and go to the enterocytes, which is then absorbed through a different pathways, either through the lymphatic system or it can directly go into the circulation system. Once it goes to the circulation system, that's where we believe it will show its therapeutic efficacy. So moving to so that was all about the background about how the lipid system works, what are the major areas where we can play around, and what we should focus on improving the in vivo viability, not just the in vitro dissolution. So if you see this slide will highlight the overall development structure, which takes from selection of a molecule to make it as a commercial success product for an industry. So when we select a drug candidate, say a known molecule already in the which is available in the form of a tablet or a capsule, we identify there are some unmet clinical needs. Maybe the drug is okay, you require 1 gram of dose to deliver due to hepatic first pass metabolism, or due to the low solubility and low viability of the drug, you require to give 2 times or 4 times of the dose. So that type of candidate selection is done, wherein while doing the candidate drug selection, you take care of the dose of the drug, whether it has a suitable log P so that it can be absorbed through lymphatic system. What is the food effect reported for that drug? What are the other PK attributes? Are there some safety challenges related to the sudden shoot in the Cmax? Are there convulsions and other adverse events of the patient reported? Fifth thing which you need to do it, is there a PGP efflux? What is causing the drug to not get absorbed completely? Or there are lots of metabolism which happen in the introsite systems, which again lead to drug not being absorbed and available at the systemic site of action. So keeping in this view, once the candidate fits in on all these 4 or 5 criteria, including PGP efflux and metabolic introsite, lots of metabolism in the introsite, that's where this candidate then move forward. When it moves forward for the pre-formulation studies, the first screening what we try to do is a compatibility study along with interaction with capsule shell or encasing. So the major challenge with lipid-based delivery is even if it is compatible with excipient, it has a tendency to, if we fill it in the capsule, it has a tendency to oil to migrate into the capsule shell. So what happens is over stability, the capsule shell tend to soften and your drug tend to migrate in the shell content. So it may or it may not give you the entire or complete drug release due to this migration. Once we find out, okay, the drug is compatible with the excipient or the lipid which we are trying to use, there is no chemical stability issues. Also, we find that, okay, it is compatible with capsule shell and the capsule shell component or the ink or the color which is used will not lead to any degradation of the product. You move it to the further development, wherein you again have a choice of deciding the dosage form. Dosage form means you can make a lipid or liquid based lipid formulation. You can make a semi-solid based lipid formulation, or you can convert that liquid lipid solution by spraying onto some inert component and make it a solid dosage form of a tablet or a capsule. So the solubility of the API stability, the what is the expected in vivo characteristics of the final finished dosage form, and the ease of handling. These 4 criteria actually define whether you wanted to make a lipid, semi-solid, or a solid-based dosage form for lipid-based drug delivery system. I'll request if anybody has any doubt, they can unmute that and ask me a question in between as well. Okay. Once you have these 4 components selected and you decide, okay, whether you have to make a liquid, semi-solid, or a solid dosage form, you move to the evaluation section. So how do you evaluate the formulation development? So first you do is a preclinical or a pre-POC study. POC is a proof of concept establishment in the industry perspective, we call it. What we do here is we make a formulation, try to administer it to a suitable animal model. By animal model, we means most probably for lipid-based drug delivery system, we choose either dog or sheep, which are the most commonly used models. Then if the formulation is showing a suitable PK characteristics, in parallel, you do an in vitro lipolysis study. These 2 studies, the POC study and the in vitro lipolysis study, gives us a confidence that the formulation is working in the desired way, in the desired characteristic style, so that we can move forward for testing in human or a clinical evaluation of the formulation. When we do the clinical evaluation of the formulation in the human trial study, we also try to check its stability as per ICH guideline, ICH Q1AR2. is the stability guidelines which generally follows for this purpose. Once we have this everything, the small scale clinical trial, or I'll say the small scale PK evaluation, simple PABE study established in the human, then we move to scale the formulation. So the initial formulation evaluation happens at the scale of say around 1 to 2 kg at max. Then we try to scale the formulation up. While scaling it up, we just not just scale the formulation, we scale the process also. So the process becomes also a critical part into consideration while scaling it up. And that's where we take care of multiple angles wherein the temperature, what, because at a bigger scale you might need to heat for a very longer time, for a longer duration. So whether it will be the stability of API will support that, will excipient remain intact in that, will shell integrity be maintained throughout the shelf life, is there a significant role of variation in humidity, like in if you supply it to the normal coastal region, the humidity would be high. If you supply your drug or the final formulation to a dried humid region, whether it would be fine or not. But that type of different humidity studies are also performed. Then we decide on the batch size, because this is important, because incomplete melting of solid excipient. So in the lipid based delivery system, there are lots of semi-solid or solid excipient which also comes in. So you need to heat them, melt them, mix them with drug before you convert them into either any of the dosage form. So when you take it in kgs, the localized heating or localized melting happens a lot of phenomena. So that becomes a critical in terms of batch size selection from an industry perspective. When you do it at a large scale, the chances of explosions or oxidation of the lipid which you do goes very high. So you take a call depending on your studies whether you require a nitrogen blanketing or normal air based processing would also help. So that is the entire most critical technical aspect at the various stages of development for the lipid. This is specifically for lipid based delivery system during the course of entire drug product development. Moving to the challenges. So what are the challenges which industry face during these 5 steps of development? So the one of the major challenges is the ability to solubilize drug, the mixture of oil and surfactant. The crucial part for these NEDs, the self nano emulsifying drug delivery system, formulation is to solubilize the API within itself and preventing it from precipitation or crashing out in the GI tract. That's the most important part, because the first part of the formulation, that solubilizing of the drug, is easier, that can be achieved with multiple combination of either oil or surfactant. But its ability to intact or keep it prevented from precipitating or crashing out is what it needs to be studied in more in depth. This is generally studied with the help of a ternary phase diagram, commonly known as pseudo ternary phase diagram, which we'll discuss further in one of the case studies. Second major challenge which the industry faces is for the self nanomemulsifying drug delivery system is the taste masking of drug along with lipid and surfactant. So somebody eating a surfactant or lipid, it gives a very acrid sort of a taste. So when we were developing this, the formulation in the industry, what major challenge we faced was the taste. Even if we administered it form of a capsule, if somebody accidentally chewed it, or if you deliver it form of a liquid only, then the bitter or the acrid taste of the lipid and the surfactant made it very unsuitable for patient to accept it. For one time of use, it is still fine. So if you use it for acute conditions, wherein simple migraine, and you have to give only one dose, and that to an occasional basis, it is still perfectly fine to have a accurate formulation slightly. But when the therapy is very a chronic term, where patient is expected to eat dosage form once, more than once, maybe twice or thrice a day for 2 to 3 weeks, that's where it becomes a lot more challenging to have a very accurate test, because patient compliance becomes an issue. So it is most important for us to have a taste mask, not just a lipid formulation which is stable, but a taste mask lipid formulation, so that it has a better patient compliance. And this is one of the most neglected area in terms of research currently. The third major challenge is the real assessment. So once we do a taste masking and all, how do you do a real assessment of whether the formulation is really taste mask or not? Because you cannot ask patient to have drug multiple times just to check whether it is taste mask or not, because it may lead to other adverse events. And there are lots of uh, ethical issues, and you have to take lots of approval. There are costs involved. So you really want to have an assessment tool wherein you can do it without exposing unnecessary drug to the humans. And the fourth challenge, which could be not, I'll not say a challenge, could be a better advantage, and if we put it like, why do you want to add an additional step of converting a lipid formulation which is liquid into a solid by spraying on inert component? If you are able to taste mask your formulation, you can directly administer the liquid in the liquid form. What you save is, you save the additional uh, excipients to be added to it. You save on the cost for the additional processing time. You save on the additional cost of downtime, which takes manpower cost. So overall, from the industry perspective, if you can have a really a taste mask liquid formulation of lipid-based delivery system, that is what we are looking for. So based on this, we have done one of a project. I can display the name for the drug as well. It's terbinafine. So what we did is we tried to develop the taste mask self nanoemulsifying drug delivery system for that. And what first we developed, as we said, first we developed this net formulation. So wherein we made pseudo ternary phase diagrams, identify the right choice of oil surfactant based on solubility studies. We also tried to do DOE studies on them so that we are sure that we had the right choice, meeting all our criteria uh, using QBD principle. So we had CMA, QTPP, everything defined for it, which we'll show in the presentation, both for formulation and the process variables. Then we also have the physical characterization done so that we are okay. Then the major component is we also did the taste evaluation. So we did taste evaluation in two parts. We did it through an electronic tongue, which is a device which have 7 sensors in them, which can measure the taste and give you the signals, and also in patients as well. And finally, the taste mask formulation was completely put on stability studies, freeze-thaw studies, various types of stress studies, so that we are sure that it doesn't crash out in the GI tract. However, human dosing was also performed for this. I will not present that data. I don't clearly have a go ahead for presenting that, so I'll keep that data safe with me. So to start with, the first thing which we did was the solubility screening. So the what was done was excess amount of terbinafine. TBF stands for terbinafine. That was along with oil surfactant and co-surfactant was taken in a vial. You keep it in a rotary shaker for 72 hours. After that shaking, you allow the vial to settle so that any undissolved particle may settle down. After that, you centrifuge again so that you have only a supernatant. Collect the supernatant, have the UV absorption done because terbinafine is UV active, and based on the calibrated systems, identify the quantity of terbinafine which is dissolved in it. So how do you characterize them? So we did a risk assessment. So the formulation, risk assessment, and everything at 3 different tools. The primary tool which we took was a heat map sort of a system. Heat map system for risk assessment is a system wherein you rate it in 3 categories: high risk, medium risk, and low risk. High risk, you mark them red. Medium risk, you mark them yellow. Low risk, you mark them green. During the development, we tried to run experimental trials heavily on high risk, tried to convert them in medium risk, and tried to run some experiment on medium risk to convert them into the low risk region. And how, and most critical of them was the droplet size determination, because you are calling it a nano delivery system. So you need, so once you disperse them, the formulation into the say the gastric media or the water dispersion system or a 0.1 normal HCl using a suitable diluent system, then you measure the droplet size of that. How do you measure that? So for measuring the droplet size, what we do is we dilute it to 100 times. Why 100 times? Because based on experience, we industry believe that the formulation is actually diluted throughout the GIT to a maximum of 100 of its original part. So if you are using 1 ml, so 100 ml is the ideal volume in which it should be added. 0.1 normal HCl is taken because the pH will be around close to the gastric pH. Whatever the resultant particle size we got, we measure them using zeta sizer. Then there are other tests also which we did, which is what is known as emulsification time test. This is done using a USP2 rotating paddle apparatus. That the equipment name Electrolab. What you do with the procedure is you take sufficient quantity, dose equivalent quantity of the mixture, add it to 250 ml of say the acid media again, and keep the Rodel partition up to 50 rpm at 37 degrees. 37 degrees for human body temperature, 50 rpm is considering GI motility. The process of emulsification, what you observe is a visual process, and what the objective is to have the minimum emulsification time so that the effect of drug can start immediately. Then drug content estimation, that is nothing but the assay in the lipid component. Then we have the taste assessment. So taste assessment, as I said, we did in two parts. The first taste assessment was done by using an electronic tongue. The lipid formulation for which the taste assessment was done with required in a 100-gram quantity. That's the minimum sample quantity requirement. You place the formulation in a 200 ml glass. The Alpha Moss electronic tray. So Alpha is the company, Amos is the brand, and Alpha Moss electronic tray is the equipment name. The system is initialized and preliminarily branched, done with sodium chloride solution. Then you keep your placebo so that you can mask all the placebo. What is the signal measurement for a placebo? In compared to placebo, you keep your lipid formulation and then evaluate your signal measurement for the final formulation, and the distance traveled by the system. I'll show you the graph later. Is what determines the taste. What the instrument shows is the smaller the distance traveled, the better the taste mass of the formulation is. Uh, the next assessment criteria was on the healthy volunteers, the subject, wherein the two formulations which were better in terms of electronic tongue data. So the placebo containing the bittering agent, so the active drug was not given, only placebo and a known bittering agent was given to the subjects so that they can have a complete dose consumed. The number of subjects were 12 and they were given an questionnaire to fill in the analysis form that how do you rate them. Now moving to the actual results, what we got from this entire study. So we'll divide it in this QBD format. So first we'll define the QTPP of them. Then we'll talk about the CQA, how we did the initial risk assessment. Then we did design of experiment. Then we'll update the formulation update variables. Similar thing we'll do for process DOA and updation of processes. So first is the quality product profile. So the target, so what was the target indication for us? So the target indication remains the same, which was it was currently used in market. What was the route of administration? The delivery route was oral. The dosage form, we targeted solution as a dosage form. Why we targeted that? Because we believe some of the patient who has tablet and capsule swallowing problem, and they may delay the drug release because you want an immediate action. So the solution would be the best choice. What is the dosage strength? The current marketed formulation, which is approved in market for this disease condition, has a dosage strength of 250 mg. We kept it at 250 mg. The storage conditions was also maintained at ICH at room temperature. We also added, this is a new component known as peculiar characteristics, that is the taste masking. So the taste masking was one of the most important quality target product profile for us because we were giving it in on a form of solution to the patient, and we didn't wanted the lipid acrylic or lipid acrid taste of the lipids to hamper the acceptability of the dosage form. And of course, then there were critical quality attributes which were defined based on QTPP. How were they defined? So critical quality attributes are one which you can test them while development, and they are most important because once you address them, the other quality attributes are already taken care. So first was the description. Why description? Because we didn't wanted the drug to crash out. So the criteria was to have a formulation which is free from visible particles. The second description was the color. So the change in color. So if the lipid component get oxidized or the drug get degraded. In both the cases, if the color of the formulation changes, would indicate there's something which is happening chemically changes in the formulation and we need to investigate it. Then the assay, of course, so that the desired dose is there. The important criteria was droplet size distribution because since we were making a nano formulation for it, the target which we set for ourselves was not more than 100 nanometer was the desired particle size. And here 100 nanometer, what I'm talking about by particle size is D90. Then taste of the formulation. So taste, we were looking for a target. So it was really difficult to quantitatively write. So we, what we do, we divided into two parts, acceptable and non-acceptable, and based on the two assessment we wanted a formulation which was acceptable to the patients. Another important criteria was the emulsification time. Since the formulation is supposed to be dispersed immediately and show action, so the emulsification time of not more than 2 minutes was established so that it gives a quicker onset of action. Moving to the drug substance risk assessment. So based on the drug substance parameters, so drug substance have, so solubility of drug will become an important criteria, which is marked as high, because if the drug itself is not soluble in either oil, water, surfactant or GIT, that will pose a lot many challenges. And of course, if the drug is very bitter and acrid in taste, again the taste of the formulation would be severely impacted. So these two components, the solubility of drug would majorly impact the description, the visual particle size, and the taste of the formulation. The other things would be majorly related to the formulation attributes of it. Sorry. So, and coming to the solubility screening, so the, as I said, the components were screened for the various oil and surfactant and co-surfactant. If you can see the bar chart depicts the amount which the drug solubilized in the various systems, Labrapac, Purolol, soybean oil, castor oil, and the glycerin 280 surfactants. So based on this solubility screening, the soybean oil was shortlisted for development. Then, as we were saying during the development, we need to have a something what is known as a pseudo ternary phase diagram. Uh, what pseudo ternary phase diagram actually includes is, it includes three vertices of a triangle, wherein at the one vertex you have an oil, other vertex you have surfactant system, along with solvent or co-solvent at the other. Here in this case we used water because water was the solvent for us, wherein we were diluting it. The overall, if you see the shaded region, is the region where we wanted the formulation to lie within. However, there are limitation to this ethnic system. So we call ethnic because it's a mixture system. So the limitation here is during the fabrication of the, when we are plotting all this page diagram, even the literature and what the excipient vendors, various suggested, these are done without the use of drug. So all these are done purely on soybean, purely water, and purely on chromophore EL and leprosol combination without the use of drug. So even in the shaded region, there could be a portion wherein post addition of drug, we can again find something which may fail. So what we were trying to say, the overall impact of drug on the physical and the chemical state was not part of this assessment. So this was not really a strong tool which were giving us an impact. Can we use this? So what we did a sort of a risk assessment based on that. So what we did, we said we have four components. We'll do oil, surfactant, co-surfactant, and diluent. Then what was the major component which we'll look for the droplet size, taste, of course, and the emulsification time. But to start with, for taste evaluation, we'll have to shortlist only two formulations. So we'll do only for two, droplet size and the emulsification time as an assessment criteria. So we designed a design of experiment. We used factors A, B, C, D, wherein the oil, surfactant, co-surfactant, and the diluent were selected. We marked them in constraint form. Constraint because the design used here was a mixture design. The good part of the mixture design is the sum addition of all four components A, B, C, D should turn out to be 1. So if you increase one component, you will have to definitely reduce one of the other component. This helps us in keeping the weight of the final dosage form constant. So once we have the design of experiment done, we also added the responses and target ranges done. So what were the responses? The responses were Y1, Y2, Y3, wherein the D90, which was a target of 100 nanometer, D50 was 50 nanometer, and the emulsification time of not more than 2 minutes was chosen. So once we had this design done, we ran the experiments and the data was put into the software. The software we used here was the JMP, JUMP software, which is a statistical software used for analysis purpose. The outcome of that software is presented here. The left graph, which is the triangle, is a contour plot, and the right one is a surface area plot, response surface plot. The overall objective of this plot is to show how the three component mixture behavior changes with different composition and how we'll get the different types of D90 or D10 particle size. So for this particular criteria, this statistical model is for Y1, that is D90. And if you see the lines, the curved lines in the left hand contour plot, it is written 50 in the small bracket, 100 in the second line, 150 and 200. What indicates it? Whatever lies within this line will give a D90 of 50 micron. Whatever lies within the line of 100 or below that will give the range of D90 of 100 nanometer, and in that way. So with what we were trying to say is, with the increase in the surfactant component, the particle size of the lipid or the final droplet size were getting reduced. Say the top vertex is oil. As we move down, the particle size goes on decreasing, and it is minimum towards the surfactant vertex C. So that's why the which is in line of the trend that the more you add surfactant, the smaller would be the droplet size. Then similar statistical model were tried to follow for the D50, wherein again both plots were considered, the contour plot and the response surface plot. And what it again suggested that here also what we saw that with increasing in oil, the droplet size were getting reduced. However, with increasing the surfactant quantity, this droplet size were getting significantly reduced. Interesting part here was, with the increasing co-surfactant, the instead of decreasing, the droplet size for D50 was increasing. That means there is a critical co-surfactant concentration which has to be maintained, not above which it will deteriorate the formulation. That has to be a cap over it. Then the third statistical model was built for the emulsification time. So the interesting part here was all the components and all the different various formulations which were tried, they all were dispersed in less than 2 minutes. So if you see the entire contour plot the various lines, it says 1 minute, 1.2, 1.4, 1.6, and 1.8. So the emulsification time was not a significant factor in this case, and all the components in the studied range would give the desired emulsification time. Now we identified the design space. How this graph was obtained is we individually studied D90, D50, and the emulsification time. We lay them one over the other. All the three contour plots were laid one over the other, and the target design range was put on it. So if you see the left side contour plot, there are two lines. One line which depicts D90 100, and the second line which depicts. So the yellow shaded region is the region wherein both the criteria of D90 of less than 100 nanometer and D50 of less than 50 nanometer is acceptable. So yellow shaded region is the only region wherein if we work, we'll get the desired formulation characteristic, and all the other gray area is the area wherein we'll get either one of the component not as favorable as we want. So based on, if you see the right side plot, so based on this, the centermost point, which is was marked, wherein the x1, x2, and x3, which is the components were looked. If you see the ratios were given, also it gives the probable predicted value of D90 at 66 and D50 of 33 nanometer, and emulsification of time of pretty close to 1 minute or less than a minute. So this formulation was shortlisted or selected rather, I'll say, based on this design of experiment studies. And if we see here, based on all this component and highly desirable zone, the formula which was shortlisted was API, because that's a known quantity, that was 250 mg as per dosage unit. The oil, surfactant, co-surfactant, and diluent in the quantity ratios as per the DOE studies. Then once we have the formula finalized, we try to move toward the various process risk assessment, wherein how mixing time and mixing speed could adversely or beneficially affect the droplet size and the emulsification time. Because processing speed will not impact the taste of the formulation, so that's why it is kept low. So majorly based on the process challenges, we'll try to see the two part, the droplet size and the emulsification time. Again, in this more simpler design of experiment was used, since we have only two components and at two levels. So we use 2 to the power 2 full factorial design, and the response remains the same. D90 as 100 nanometer, D50 as 50 nanometer, and emulsification time is not more than 2 minutes. When this all the experiment was studied and data was populated in that, if you see this four component, the upper left hand corner, the graph shows a plane which is pretty much in parallel to the ground plane. Even graph 2 and the graph 3, which is left side bottom graph, which indicate the impact of mixing speed on D50, D90, and emulsification time, is pretty much the same. Then we what we were trying to see is, we at the right hand corner we have put a Pareto chart. So do any of the component have a significant impact on the process formulation or not? So if you see, there are two lines which are marked in the Pareto plot. One is the black line, one is the red line. Anything which is below the black line, the Pareto chart, doesn't have a significant impact on the formulation. So all the three components, 1, 2, and 3, which was studied, didn't have any impact significantly. So what it means is, no matter at what speed, at what RPM, what time we do, there is minimal impact on the formulation variable. So that is something which is the most desirable in terms of industry. Why? Because the scale up parameters will become invariably very easy to scale the formulation from lab scale to the commercial scales. And process is very robust enough to give a consistent result throughout the development. Then once both formulation and process were established, then we move to how do we taste mask it. So for that, as usual, we do flavoring and sweetening agent combinations were tried. What we did three flavors: cherry, orange, and strawberry. And sweeteners were four: aspartame, acesulfame K, sucralose, and monoammonium glyceride. Majority portion, if you see the first line, the TBF optimized nets formulation, that is 98.6%. In the first six component, and the last two, it is like 1% less. In the so major component of this entire formulation would be the four formulation which we have just optimized. It would be additional 1 to 2% which we are trying to add flavor and sweetener just to taste map the entire formulation. Now we can discuss about why we chose only cherry, orange, and strawberry as a flavor. Cherry as a flavor has a tendency to give to mask oily taste of the drug. So cherry, banana are the only two flavors which are known to mask the oily component or oily taste in the tongue. Orange flavor is what gives a biased, what we call is a biased flavor. Why we call it a biased flavor? It try to divert the mind from the oil type of a taste to a orange type of a taste. Strawberry flavor, why it was chosen in that fashion is it was also suggested similar to like cherry flavor, that strawberry flavor would also mask the acrid taste of the oils. The sweeteners here used are not are the high-end sucrose, which have very high sweetness in terms of sucrose. So that very small quantity would make the taste very sweet. So this is the taste assessment result of an electronic tongue. I'll again brief the electronic tongue here. So this is the major component of the entire content. Electronic tongue, what it has is it has seven sensors. Seven sensors, when dipped in the formulation, they give the electronic signals. When these electronic signals of the seven sensors placebo versus the final formulation is compared, it gives a difference. That difference is actually measured in this bar graph in terms of distance. This distance is distance traveled by the signal with respect to the placebo of formulation 1 to 8. The minimum of the minimum the travel, the better the taste masking. So the TBF6, if you see the formulation number 6 is one which has the minimum distance travel, maybe around less than 400 or close to 400. So that was the best taste mask formulation which was taken based on the electronic tongue bitterness masking model. There are different model in the electronic tongue. So this model suggested us TBF6 to be the best one. Then what we did with, if you see here, next to the formulation 6, there is one more formulation that is 7, which is second best. Though it is more, 4 and 7 are pretty close, but 7 is even better. So the two best formulation as we defined earlier, we'll take it for the screening in the human. So what we did, we took formulation 6 and formulation 7 both, and this is the assessment parameters of the 12 subjects which were given to them. Um, the green one is for, which is highlighted, is for formulation 6. TLC stands for placebo because no active drug was given to any of the subject. Equivalent quantity of bitterness agent was definitely added to the formulation, and the various assessment parameter was, as soon as you add the component, what is the initial mouthfeel, is the sweetness of the formulation good enough, what is the bitterness of the formulation, is there something which comes as the aftertaste once you have taken the formulation and you have a sip of water, after 2 minutes do you feel there is an aftertaste of the formulation, and how do you rate the overall mouthfeel. So based on this assessment parameters, again the formulation 6 which we got from the electronic tongue as well, even the human subject accepted that that formulation is far better than the next best formulation, that is formulation 7. So based on the electronic tongue and the human studies, the final formulation which were derived was this component, wherein terbinafine, the first 5 component is about the core SNETS formulation, the last 3 cherry, aspartame, and monoammonium glycerinated combination, which is for the taste marking purpose. Then we move to the stability studies. So that TBF, the final formulation was put on both as per ICH condition, accelerated and the long-term stability study. Uh, Description, assay, particle size, emulsification time, and other than that, related substance and many other components were also studied, which are not populated it here. The major what we want to show is the particle size. The over the stability of accelerated or the long-term condition, the major component of particle size and the emulsification time remains unchanged. So we were able to retain the nano emulsification property of the formulation during the ICS stability condition as well. Then we do a one sort of a freeze thaw study. So this is a stress study. So when we transfer the say the final formulation from here to the other countries, maybe a cold country or from in currently the northern region of India, wherein the where there is very cold. So the final formulation may be exposed to various conditions of extreme heat and extreme cold. So we do a freeze thaw study to see that the formulation should not crash out with that. Usually the protocol says to do 3 cycles of freeze thaw study. What we do is you keep it at the room temperature for 24 hours, then first freeze it to so that the entire component is freezed at 2 to 8, then thaw the sample immediately to 40 degrees for 24 hours, then immediately freeze and 2 to 8 for 24 hours. So this way freeze thaw is done for 3 times, and at the end of the third thaw cycle, you keep the sample again at room temperature for 24 hours and then analyze. So the first, so we have analyzed it first cycle, first freeze, thaw, and third cycle both freeze and thaw. So any of the freeze or thaw condition doesn't really impact the formulation. So so the formulation per se is very stable and good in terms of all handling and logistic challenges during distribution of drug. Based on all these studies, we updated the risk assessment since we initially did some risk and we modulated the risk. So we need to mitigate them also. So based on the studies, the star mark here on the various components are the risks which we have reduced based on the study from the previous mark studies for drug product. For formulation variables also, since we have studied that emulsification time, we have also optimized the taste of the formulation. We have also done the droplet size distribution studies on the stability and description and physics. Visual observation of the stability samples also didn't show any change. So I think the overall formulation risk is also low, and which we have mitigated through a series of experiment and trials. Process, definitely we have studied there is no impact of any mixing time and mixing speed, which is very good for the scale up and making it a commercial formulation. Last to summarize, we were able to make a unique TBS SNET formulation. TBF is terbinafine formulation, which is a very poorly soluble drug. We also established an electronic, which is a very good alternate for us to do a taste mark studies, which was a expensive study in terms when we did it for human trial. Maybe for future we'll just rely on the taste mass electronic tongue data to move forward for uh, to have single shot study for the taste mass formulations. Just to again re-emphasize what we see is SNET's approach cannot be applied to highly soluble water soluble molecule. This is purely restricted to the water insoluble or a highly hydrophobic type of molecule, because there are other simpler approaches available in industry or in market currently, which can be utilized without venturing into this high-end complex formulation. Compatibility is of oil and surfactant in the GI tract is still which we need to do a study, and how long-term administration of this oil and surfactant mixture to human will impact is still a gray area, and studies need to be done in that area. If you deliver, say, for a chronic disease, this medicine is given for, say, around a period of 2 months or 3 months. Does it have a significant negative or a positive impact on GI flora, GI lining, and other adverse events? So that is yet to be studied. One of the this is the third point which I want to highlight. I have seen many formulations, very good formulations based on SMET formulation, which is available in public. Very example is for silicoxib, which is there are many SMEDs and SNET formulation which is made. But the issue in industry, what we face is all this research is done in a very suboptimal dose. So what is done is celecoxib to be patient has to be given in a dose range of 200 mg. But what the formulation of the article which has been published in the range of 5 to 10 mg. So even if you have a very good formulation and which you have make a very good and you show it is working efficacy good wise in terms of animal model also, uh, using it at a very suboptimal dose make that literature very not so good for us industry, and it gives us a sense that it is difficult to make a bigger formulation with a higher dose. So my request to all the researchers here would be, please look into this. We generally and usually we should try to take the therapeutic dose which is readily available at US FDA side, even in the India SIMS and MIMS side, at what is the dose to be taken, and not to study much on the suboptimal dose of the drug. The another challenge or which I see is the total daily intake of the excipient use here, not just the drug. So all the components which are oil surfactant which are used in SNETs, MEDs or other lipid-based formulation, they have a certain limit above which they cannot be used. Or rather, I'll say the studies to that extent that they can be used have not been performed. As the industry matures, as more and more studies will be done on this excipient, even the excipient vendors do a lot of research on animal model toxicity studies. As more data pull in, we'll be able to do much more better in terms of lipid delivery going forward. But for the research purpose and we'll suggest, will request from industry point that will not take anything which is more than permissive. So even if you make a very good formulation using something known as which is way beyond which a normal healthy human volunteer can take, that formulation is of no use because it will ultimately not never make to the ground. So patient will never consume it. It will just be a research. So my request would be to look into the IIG limits. So how many a times I get questions that how do we look into the total daily intake? So you can Google it out to go to US FDA side. There is inactive ingredient side wherein you can put the name of the excipient. Therein they'll give you the what is the maximum daily intake of that particular excipient above which it is not rendered safe. Below that, it is rendered safe. So use the components, whatever you use in the formulation development within that criteria, within that limits only. These are the few references which I referred during the making of this presentation. I hope that this references would be helpful to you. These are few are latest, few are old. There may be other better publications also which you can come across. Please go ahead if you can find out. If you find something which is very good, share across with me as well. On this, I'd like to have give a special thanks to both the professors and the entire team of QIP and KLE Belgavi for giving me this opportunity to present and interact with you. Now I'll open the forum for questions if you have any.
Thank you, sir. Sir, there are some few questions.
Mm-hmm.
It's not one. So there is a question from Shweta. Kindly let us know some source for electronic tongue evaluation and approx cost per sample.
Okay, so thank you, Shweta, for that question. So electronic sample, so there are two electronic tongue which is available across the world. There are two different models. One is a Japanese model, which is Alpha Moss, which I presented here. There is one more in cent base. So in India, you will find both this component. One is it in IICT, I believe, and what you can also do is you can write to the equipment vendor as well. So they are open to do the analysis and send back. So if you get a letter from your institute and send it to the vendor of the electronic tongue model. What you to do is, whenever you present or you publish your article, you have to give reference of that you have used their electronic tongue equipment for generating the data. I am sure they'll support you for that.
Sir, can you please repeat the source again?
Okay. So electronic tongue model, it is available with the equipment vendor.
Okay, sir.
It is also available with IICT. But what
Okay,
I'll
sir.
suggest is go
Yes, sir.
with, because the other institute may charge you. You can
Okay, sir.
contact to the, you Google it, you will get the equipment manufacturer India site.
Okay,
You
sir.
write to them the mail, they'll give you the and send them the sample as per request. What in
Okay,
turn
sir.
they'll require you to do is, whenever you do a publication using this data, you'll
Yes, sir.
have to acknowledge them saying that you have used their data as a routine practice what you do.
Yes, sir, definitely. Sir, all this time, actually, we used to have the electronic tongue, which is there from tea estates and all, which they will be using. That's what we have used.
Mhm. So do you find that that is good?
It is okay, but still, if it is a better one for the beta drug tasting, then thought maybe the authentication will be much more better, sir.
Yeah, so what I'll
We,
suggest is, for electronic tongue, actually there are two different models. So
yes, sir.
what it
Yes.
comes with is, one of the, so one instant ka jo hota hai, that gives only 5 taste,
Okay, sir.
and that gives the combination of taste.
Yes. Yes,
What
sir.
alpha T alpha mass has is, they have 7 different probes. So they
Yes,
have
sir.
a
Yes.
different probe for bitterness, different probe for Unami, different dose for acrylic. So you
Yes,
can,
sir.
so combination of both you can try. So
Definitely,
what
sir.
I'll suggest is,
Definitely.
if you really want to do a good research for your formulation,
Yes,
you
sir.
can do your formulation evaluation on
Yes.
like both of them, and put a comparative study, so that it becomes easy for the future generation to have which one to use based on your research.
Yes, definitely, sir. Thank you so much, sir.
Sir, am I audible?
Yeah, sure.
Yeah,
May I
sir.
proceed?
One more thing, sir. What type of samples can be tested? For example, if we know the drugs can be sent, yeah, because you have told that we can send it to the vendor. So, do you have to prepare the sample before sending? So, for example, if I want to get a drug-loaded lollipop tested for its bitterness, whether I have
Mhm.
to send the lollipops or I have to make the solution of that and then send?
So for that, what I'll suggest is, if ultimately the lollipop has to dissolve and then give the taste, right?
Yeah.
So it would be, so you make the lollipop, you can give the crushed sample of the lollipop to the vendor, because
Okay.
they'll, because vendor will not try to do the making
Some,
a
yeah. Yeah.
volume maker, because they'll not try to invest their time in that. You can
Okay.
crush the sample and send along with them a vial of water or whatever the component you want. Ask them just to mix immediately and give a suitable time within 2 hours or within 4 hours. Please run the analysis and give it to you.
Okay, sir, please help up with one or two vendors who can who actually does it as a gift sample or something like that.
So, okay, so what I'll do is post this. I don't have currently ready-made with me, but I'll share the email ID of both to whom we generally write, and I'll
Okay,
request him personally on phone also, to so that whenever you send him a request, he can actually get your sample done.
thank you, sir. Thank you very much.
Sir, there is a question from Virendra, which is the reliable software to design pseudotinary phase diagram.
But never knew that vendor also does it.
Okay. So just to answer that, when you don't rely on a software, the best thing is you can actually do it using a graph paper. Why I'll say that is because that will enhance your. So first you do it on a graph paper. That will enhance your way how you put it, how do you interpret it, and how you design it. Coming to your original question of only a software, JMP software, or glass, that will be an expensive one. A simpler one will be a glass prisma. If you, I don't know whether you know about it or not. Glass prisma is also a software that is readily available. uh, that is, that can also make the pseudo ternary phase diagram. But if you want the most authenticated one, which even the auditors and the FDA and the big regulatory authorities use, so that is the JMP software. That is a SaaS based software.
So there's one more question from Virendra. Why not zeta potential and transmittance of SNEEDDS is not considered as a quality critical attributes?
Okay, so zeta potential, and that's a very good component. So zeta potential ultimately what will it shows it? It shows the stability of the formulation. So zeta potential becomes important in terms when you have a nano suspension sort of a criteria wherein the suspension form is to be stabilized over the period of stability. Herein, the advantage of self nano-emulsifying delivery system, it is a clear formulation. It forms a nano-emulsion only when the patient takes it. So actual the emulsion formation is inside the body. So if something is happening inside the body, the zeta potential will be impacted much by the GI components of the individual patient, and that will vary a lot. So really zeta potential will not be a criteria in terms of self nano and even self micro emulsifying drug delivery system. Hope I answered that. If she has further to that, I can still discuss.
So there is a question from Samradni. How to
Mhm.
set the ratios of oils and S-mix in pseudotinary phase diagram? And could you put forward that is there any method other than water titration method to select the optimum ratio of oil surfactant and co-surfactant?
So thank you, Samridhini, for that. So what I'll suggest is you don't really to make the pseudo ternary phase diagram. You can write a vendor name known as Gete Fosse. He is one of the major vendor who supply lots of lipids, various lipids and oil. Gete Fosse at its site has this pseudo ternary phase diagram for its component already made for you. So we industry requested them that we don't want to waste time on that. So they have displayed it for all in general. And so they have all the pseudo ternary phase diagram readily made. But only problem or the pain point for this pseudo ternary phase diagram is they don't have your drug component in it. So you need to choose based on the pseudo ternary phase diagram, which diagram suits you, and that particular S mix and oil component, the center point you can take and add your drug. That becomes your first point criteria to check. If that optimized formulation in the center point is failing to meet your criteria, then that is not the right choice of oil and S mix. You go for the other one. So what I'll suggest is rather than spending time of what is already done, to go to their website, get all the pseudo ternary phase diagram, add your drug component to the optimum ratio of their diagram, see whether it works or not. If works, that is the suitable oil component surfactant ratio for you. If not, try other one. You'll have to experiment a few. Maybe in a say 4 to 5 experiment, you may be able to arrive at your optimized formulation. And in case if you want to do on your own, I'll suggest you run a DOE, design of experiment. And for that, you can use any of the statistical software, maybe JUMP, maybe Minitab, or if required, design expert from Stratis also would do. Do a mixture design. Mixture design would help because it is similar to the pseudo ternary phase diagram, and that will give you the components.
So there is a
Sorry, I
question
missed.
from Anant Bhandarkar. What is the role of droplet size and zeta potential on stability of SNEDDS?
Okay, so thank you, Anand, for that. So again, I'll try to emphasize why the role of droplet size. Droplet size become important because even if you give so we don't measure the droplet size of the formulation. We try to dilute it in 0.1 normal HCl, considering it's in the stomach condition and pH. Would it still be able to remain the drug in the nano size? Second, it will not try to crash out the drug. So drug doesn't become separate and the oily component doesn't become separate. So droplet size is definitely very important because that will determine whether it is really a SNEDs or not, whether it's a stable SNEDs or not, and whether it will form a micellar structure which is very quick and it will be absorbed quickly. Again, I'll repeat the same for zeta potential. I say zeta potential is generally required to stabilize a nano suspension on a nano emulsion formulation. This is not a nano emulsion formulation. It is forming nano emulsion in vivo. in the real terms, in a tabletop, when you will administer and give it to the patient, it will be a clear liquid sort of solution. So that's why it is self nano emulsifying. So the moment you add water, it will automatically emulsify. It as a component, it is a solid. If you ask me in a layman term, not very scientific, it is a solution of oil, surfactant, co-surfactant, and drug, completely devoid of water. And that when added to water, convert to emulsion. So that's why data potential doesn't play a significant role, because all you want is for 1 or 2 hours before the drug get absorbed.
So
Anand, did I answer that?
Anand sir has one more question. What is the
Okay.
range of drug loading capacity in SNEDs was considered as ideal?
What is the range of drug loading capacity?
What
So
is
I got that. So the range of drug loading capacity, it is difficult to say. Why I'm saying is, for our case study, we defined it based on the therapeutic dose of the drug. So since the drug dose has to be 250 mg, and to have a therapeutic efficacy, you will have to administer 250 mg. That's why 250 mg was chosen. If you ask me in general, what could be the drug loading capacity, that depends on individual drug to drug molecule. That is difficult to say. Only thing I can say, surely say is, which I highlighted earlier also, is don't go to a suboptimal therapeutic range doses just to make a good formulation. If that therapeutic dose, if this net is not working, look for some other component. But at least work on the therapeutic dose of the drug.
So one more question. Please elaborate on preclinical study for POC establishments.
Okay, so for the preclinical study, okay, what we did was, you take a couple of there are different models which are available. How do I explain to that? Okay, so once you have the preliminary formulation made out and you keep them ready, so you wanted to test it. For industry, going it and doing a human study is very costly affair. There are lots of ethics committee approval, and you yourself want to be sure because you are dosing to a human. So considering all that, you do try to, for this type of complex system, you try to do some sort of a preclinical study, which we call a proof of concept. How do you establish the proof of concept? Maybe you take a everted gut sac or a different system, put this your formulation in that, check whether the cell lining which is there, it doesn't die out, it doesn't show a significant change in morphology. It depends on the type of study. It's a very vague to say what type of preclinical study, because it depends on the objective for which you want to do the study. We did almost like 7 preclinical studies before we established this proof of concept. If you ask me in terms of formulation wise, you can, like I said, you can do a small animal model. If you don't want to do that, you can do like, don't use rat, because rat is not a suitable model for lipid based system. Dog surely would help. Dog is a good model for lipid based delivery system, because that GI enzyme system is very pretty much close to us, and that's effective one. Sheep is even better, but sheep is a costly model and it is very difficult to find in India. So dog model is still a better. Rat people do occasionally at industry, the academic level, but for that, what I'll suggest is that is only for acute dose and for some small dermal studies. Because it doesn't have a complete GI system and track close to human, what we are, that will not give you much of an information.
So there's a question from Sabha. She's working on nano formulation. She's having difficulty in getting nano size range. I would be glad if you could guide me in getting nano size less than 200 nanometer.
So I have a question for Sabha. So is she making a nano suspension or what?
Yes,
Nano formulation is a very big term.
I'm making a NLC. NLC, sir, nanostructure
Okay,
lipid carrier.
nanostructure lipid
I
carrier.
have got size. Yes, sir. I have got a size of 400 nanometers, which I'm using it for oral delivery, sir. So I wanted it to be, in less than 200 nanometer.
Okay. So, Sabha, a few more detail would be required from your side. So, while making an NLC, are you using a probe sonicator?
Yes, sir. I'm using a probe sonicator.
So, can you detail me the process of probe sonicator
Yes,
make model?
sir. Actually, sir, I stay in Pilen only. If required, I can come and meet you, sir, with the data.
So, what I'll suggest is, Sabha, I'll drop my email ID to the. You can drop me a
Yes.
mail for the detailed procedure because I'll need to go through the
Yes, sir. Yes,
background,
sir.
read some background, and then I'll suggest you for that.
Yes, sir. No problem, sir.
Okay, thank you.
Okay, sir.
Sir, there is a question.
Thank you, sir.
I didn't heard. Is there any other question?
Sir, I think hope all the questions have been addressed. So coming to the end of the session, of course, it was a very interesting, comprehensive information on SNEDs with special emphasis on critical quality attributes and risk assessment parameters, evaluation aspects, special emphasis given about the taste masking component for SNEDs, the industrial challenges and the future scope. Thank you, sir, for this very interesting session, and it has helped many young minds they have got answers to their questions and troubleshooting problems. Thank you very much, sir. On behalf of
Thank you, Professor, for giving me an opportunity. To be honest, sorry for cutting you in between. I like to speak to all the participant professor here. What my mentor in industry taught me is the process of one-way learning is now over. Only we can progress if it is a two-way learning. The question you ask me make me also think more so that I can also learn more. It gives me new ideas. If you have idea, feel free to write me. We'll connect. I'll connect to you. You don't need to connect to me. If I like your idea, I'll connect to you. We'll work together for it.
the delegates can always, we have your mail ID and your contacts. If any delegates are interested, they can get it from us and get in contact with you. Thank you very much.
Thank you.
Again, on behalf
Thank
of
you, everyone.
AICTE, thank you very much. So you can leave the meeting so that we can progress to the next session.
Sure,
Thank you.
thanks.
So dear participants, we have concluded with the first session of the QIP. We have another interesting session which is commencing at 11:30 by Jacqueline ma'am. She's from Ajanta Pharmaceuticals. So please join in another few minutes of break. After taking a few minutes of break, we start the we'll be starting at 11:30 the session. Thank you.
Madam,
Shall we go there?